So now press my buttons. So hi and welcome to this webinar in the FibRe webinar series that we're making, that we in Treesearch are making together with the Competence Center FibRe. So we have a speaker today, of course, is César Själsjö from University of Gothenburg and Swedish NMR Centre. And he will get a proper introduction by Professor Annette Larsson in just a minute. But before that, I just want to say that if you have any questions, and please have questions of the speaker, we will have a Q&A session after his presentation. So if you are in Zoom, you can just raise your hand, type a question in the Q&A, or whatever, make us notice you, and we will let you ask the questions. If you're following on YouTube and have a question, you can just type it in the chat, and we will be sure to check the chat there too. So with that, I give the word to Annette.
Thank you, Josephine. So my name is Annette Larsson. I'm the director for the Competence Center FibRe. So and we are so glad to do this together with Treesearch. And today we are also very glad to listen to a DNP NMR presentation by César Själsjö. And you were you were the first PhD student from Professor Emsley in Lausanne. So you are probably one of the most experienced DNP NMR persons in Europe. So we are really looking forward to your presentation. So please go ahead.
Thank you, Annette. Thanks for this nice introduction. So yes, my presentation is about how we can use DNP NMR to characterize cellulose. I know that what I do can be sometimes a little bit obscure and hard to understand. So I usually try to make my presentations in a very pedagogical way. So my goal here is really for everyone to understand how it works and what type of information we can get from DNP NMR about cellulose-based materials. I usually start with an introduction of myself. Annette did a little bit of the work. But so despite my really Swedish sounding name, I am not Swedish, I'm French. I was born in the south of France, in Toulouse. I did most of my undergrad studies there. And then I moved to Lyon, where I discovered NMR, nuclear magnetic resonance. I was working at the ENS school in Lyon. And because I loved NMR so much back then, I decided to do my PhD in Switzerland with Professor Linden Emsley at the École Polytechnique Fédérale de Lausanne. So I already started to do DNP NMR for 4 years of my PhD. And then at the end of my PhD, I decided to do more DNP NMR. I moved to Copenhagen at the Danish Technical University, where I did some more DNP NMR, a little bit of a different topic, but still DNP NMR. And I did that until 2020, where I got recruited as a research engineer at the University of Gothenburg. So I've been working here at the university since 2020, and I'm a docent now, so I'm doing research on materials, cellulose materials, of course. I also do a lot of pharmaceutical formulation. I have a collaboration with AstraZeneca, where I use DNP NMR to characterize their pharmaceutical formulation. So this is the lab where I'm working and where I'm giving this presentation from, Swedish NMR Centre. We have essentially a building just to ourselves with a very large hall where we can put all of our magnets together. And this hall has 2 magnets that are very useful for studying pharmaceutical formulation and cellulose-based materials. I will enter into more details later. So initially, I was trying to describe why we would want to study cellulose, and then I figured that probably most of you know that 100 times better than I could ever do, because I'm not a cellulose chemist. I'm just a physical chemist who's using NMR to characterize a lot of different products. So I'm not going to try to convince you why cellulose is really interesting to study. I discovered myself that cellulose was very important when I arrived in Gothenburg. I saw that a lot of people at Chalmers, including Lars Evenes, was using NMR to study cellulose. So that's how I initially got in. So since I arrived here, I've studied different types of cellulose. And the first type of cellulose that I have been involved with is dialcohol cellulose. So the idea is that for dialcohol cellulose, we start from regular raw cellulose, and we break the bond, the C2C3 bond, to create a aldehyde, and then we reduce those aldehyde into alcohol. And the idea is that we do that because even though native cellulose is very strong and sustainable, it's not really easy to process like plastic is, for example. So the reason why we do dialcohol cellulose is that because we make these changes into the backbone, this cellulose becomes much more easily processable. And so then we can use that to create some thermoplastics, which are renewable, such as those 3D printed material. And so when we do those types of renewable material and we do this modification on the cellulose, we need to be able to know to which extent the raw cellulose has been converted into dialcohol cellulose. So that's one of the questions that we want to answer with NMR. The second type of cellulose that I've been looking at is ethyl hydroxyethyl cellulose. So it's essentially some cellulose where some of the hydroxyl groups on the cellulose are replaced with ether groups. So either methyl, ethyl, or hydroxyethyl cellulose. And these modified cellulose are used for paint, for example, or for a nail polish. And depending on what amount of substitution there is and where the substitution is on the molecule, the property of that cellulose will change because it will be more or less exposed to microbial contamination, which will lower the viscosity. And we want to be able to control the viscosity of the paint or the nail polish that we have. So because of that, we need to understand what type of substitution pattern we have on the cellulose. And one last type of cellulose that I've been involved with is HPMC, hydroxypropyl methyl cellulose. This type of cellulose is essentially a cellulose where we replace the hydroxyl group with a isopropyl, hydroxy, and methyl cellulose. And this hydroxypropyl methyl cellulose is used for pharmaceutical formulations. So for example, if we have a poorly soluble drug, we can mix it with hydroxypropyl methyl cellulose, and then it will improve the drug's dissolution. Because most of the time when we have a drug, we want it to be able to dissolve rapidly. Also, if the drug, let's say it's amorphous and we want to prevent it from recrystallizing, then adding hydroxypropyl methyl cellulose, it is helping to prevent this recrystallization. So this HPMC is used as an excipient together with the APIs to essentially be able to control the two properties that I've described, but much more than that. So I'll show you also how we characterize HPMC. So in general, for cellulose, we want to be able by NMR to say, okay, we've modified those cellulose backbones, but where did we modify it? And by where, I mean where in the molecule, but also where in the particle, because we have, when you have a powder, you have particles, and you want to be able to know if the modification you've done has been done only at the surface of the particle or inside or homogeneous, because this will change the property of the material. You want to know how much it has been modified and how evenly. That's what I just described. So for that, we essentially need some kind of an MRI machine, but the difference is that instead of putting a human, we're going to put cellulose in it. So an MRI machine, you know, it's called MRI because it's magnetic resonance imaging. So you put a body in a magnet and you get an image that the doctor can use to give you a diagnosis. And the idea, what I have been doing for more than 12 years now, is that we put cellulose inside this big magnet, and then we don't get an image, we get what we call the spectrum. And that's why it's called nuclear magnetic resonance instead of magnetic resonance imaging. And because we put something that is solid, it's called solid-state nuclear magnetic resonance. I actually did a video where I'm showing you how I'm preparing the samples for solid-state NMR. So here it's not cellulose, it's a drug that I got from AstraZeneca. And I put this drug in this tiny little cylinder that's 3.2 millimeter diameter. So I only need essentially a few milligrams of the pharmaceutical compounds. And when I do that with cellulose, it's the same. I only need between 30 and 40 milligrams. And then when this cylinder is full of my material, I go to the magnet that is the best to do either cellulose or pharmaceutical formulations. And I put this tiny little cylinder at the bottom of the magnet so that it gets inserted into the middle of the magnetic field. And then I just go to the computer where I can record the NMR spectrum. So this is what a typical NMR spectrum would look like. Here I take the example of thymol. It's this molecule, and I show you the hydrogen NMR spectrum. So what is nice with NMR is that each of the hydrogen atom of the molecule, they have a different peak on the spectrum. And so this is very convenient because we can know which peak corresponds to which hydrogen, so we know which type of hydrogen is present in the sample. This is really nice, and this type of spectrum, this x-axis here is called chemical shift. This chemical shift, it's essentially dependent on the electron density of the nucleus we're looking at. So before I was showing you hydrogen, but here I am demonstrating how chemical shifts work with carbon. So the idea is that if we have, let's say, a carbon atom that is next to those electronegative elements will pull out the electrons away from the carbon, and those carbon will have less electron density around them, as opposed to some other carbons that have different neighbors that are less electronegative than oxygen. And in that case, the electron density will be a little bit bigger. And this electron density will be what makes the peak showing up at a different place of the spectrum. So if I take this molecule and I record the carbon NMR spectrum, then the CH2, this is glycine, the CH2 will appear at a lower chemical shift value because it has a high electron density, and vice versa, low electron density corresponds to a high ppm. So like this, it's very convenient, even with a molecule with a lot of different carbons, because each carbon will not have exactly the same environment. It will not have exactly the same chemical shift. So we will be able to distinguish each carbon with its chemical shift. We say that chemical shift is a fingerprint of the molecule. So what about cellulose? If I want to do carbon NMR of cellulose, I'm going to get something that looks like that. So the difference that you probably notice between this NMR spectrum and the previous one that I showed you is that here the peaks are broader. And this is because the previous spectrum that I showed you was recorded in liquid. And when you have a liquid state NMR spectrum, the peaks are much sharper than in solid, like here. We say that we have a low resolution when the peaks are broad. And so because the peaks are broad, you can see that some of them are still well defined. For example, the carbon peak of the C1 of the glucose ring is well defined. Same for the C4. But you can see that here, the C2, C3, and C5, they are all overlapping onto each other. And this can be a problem sometimes, especially if you want to look at what's happening on the C2 position. I will show you later why this can be a problem sometimes. And the C6 is thankfully resolved enough. And this is a solvent that we use. I'll come back to this later. But so because solid state NMR has low resolution, this can sometimes be a problem. And on top of having low resolution, solid state NMR also has low sensitivity. So the resolution is about how broad the peaks are, and sensitivity is about how high they are. And so, for example, if we look at carbon NMR, the isotope of carbon that we're looking with NMR is carbon-13. We cannot look at carbon-12 because it doesn't have a spin. So we can only look at carbon-13, and the natural abundance of carbon-13 is only 1%. So we're essentially only looking at 1% of the sample. And this is reducing the signal by a factor of 100 compared to if we look at proton or hydrogen, I should say, because the proton are almost 100% abundant naturally. But we have other problems with proton. So in solid state NMR, I will essentially never show you a hydrogen NMR spectrum because they're so broad that they're all overlapping with each other, and it's impossible to tell which peak is from which hydrogen. So for the rest of this presentation, I'll show you some NMR spectra of carbon-13. And so there is low sensitivity because of this low natural abundance, but also because the peaks are broad. So here I'm showing you the example of two peaks. They have the exact same area under the peak, and one is broad and the other one is sharp. And so the area is proportional to the amount of carbon. So here I'm essentially comparing one carbon in solid state and one carbon in liquid state. It's the same amount of product. And so the same amount of product will result in a much clearer peak just because the peak is sharp. So on top of having only 1% of carbon-13, because the peaks are broad, it gives us an even smaller signal compared to liquid state NMR. And this signal is hard to detect over the noise because we have a noisy baseline, because when we acquire the signal, we do that with electrical cables. And those cables, there's some thermal signal that comes with it. So it makes it hard for us to detect the signal over the noise. And so because of this lack of sensitivity, we need to acquire the measurement for a very long time. Up to, let's say, the 1990s, solid state NMR was restricted to either very long experiment or not carbon because it would be too low sensitivity. But since the 1990s, there's this new technique called DNP, which is what I've been doing since my PhD. It's called dynamic nuclear polarization. So the idea is that we want to increase the signal, and DNP is a way to increase that signal. And so to increase the signal of solid state NMR, we will essentially, on top of, let's say this is the powder that we want to analyze, we will impregnate this powder with some liquid. We choose carefully the liquid so that it doesn't dissolve the solid that we want to look at. Otherwise, we're essentially doing liquid NMR. So we add what we call a non-solvent, and we have, let's say, 30 to 40 milligram of powder, and we'll add 20 microliter of this solvent. And we mix those two things together and we get something like that. It's a wet powder. And then we put that wet powder into the cylinder that I've showed you on the other video. And so if we do that for cellulose, we would essentially have the solid, let's say 30 to 40 milligram of cellulose, and then the solution that contains radical molecule. So we're going to call this solution a radical solution. So it's essentially a solution that is usually water-based. At least for cellulose, almost all the time I use water. And then on top of water, we'll also use a glassing agent. This glassing agent is either glycerol or DMSO. And the reason is that the radical molecule that we will dissolve in that solution, it's very low concentration, by the way, 12 millimolar of this radical molecule. You see the unpaired electron on this bi-radical molecule. We want those radical molecules to be homogeneously dispersed. And so when it's a liquid and you dissolve the radical in that water glycerol mixture, it will be homogeneous. But the thing is that we can only do DNP at the very low temperature. We do DNP at about 100 Kelvin. So all the spectrum that I will show you are recorded at 100 Kelvin. So even if we add a liquid solution at the end, we freeze that so we still analyze a solid. And we use glycerol and DMSO because we want that during this freezing process, we want to prevent any crystallization of the solid and liquid mixture. If we were putting pure water, the water would crystallize and then it would exclude the radical from the crystalline domains. So we would have pure water crystalline domains and then radical aggregation. So we wouldn't have a homogeneous 12 millimolar concentration of the radical everywhere. This is very crucial for DNP. So this is why sometimes we use a glassing agent like glycerol or DMSO. So yeah, the radical solution must be amorphous when we freeze. It needs to dissolve the radical molecule. And in our case, sometimes with cellulose, the cellulose is actually acting as the glassing matrix. So sometimes we can just put water and the radical inside the cellulose, and the cellulose will essentially be swollen by this radical solution. And then this swollen matrix, when we freeze that, it never crystallizes because it's a homogeneous mixture of water and cellulose. So then we don't even need glycerol, which is even better because when we do carbon NMR, glycerol has carbon and DMSO has carbon. You've seen that on the previous spectrum. And so we have some contamination peak that we're not interested in. So it's good if we can avoid having to use those glassing agents. And so this is what we get essentially when we do everything correctly. In blue, you have the carbon solid-state NMR spectrum with DNP. And in orange, I compare that with the same material, but without the DNP effect. And you can see that we increase the signal by a factor of several tens. So in the following, I'm going to have to separate two cases. The first case is when we add the radical solution and the cellulose is, the solution is swelling cellulose. So we have this expanded matrix with water and eventually glycerol and DMSO if needed, and the radical homogeneously dispersed in space. And so when this is happening, the signal is enhanced everywhere by the same amount because it's all homogeneous. So in that case, you have all the peaks that are enhanced by the same amount. And you can calculate this enhancement. We usually call it epsilon. It's the ratio of the intensity of the signal when we use DNP and we don't use DNP. And in that case, if we do things correctly, we have something, in that case, it was 70, but sometimes I can get more than 100 enhancement. So the signal can be increased by 100 with this technique, which is really crucial if we want to do some experiments with a low amount of product, or I have some pharmaceutical formulations with low amount of cellulose in it. We really need this enhancement of 70 or 100. And so, like I said, all the carbons are enhanced equally. And so we've used this technique where we can swell the cellulose thanks to the radical solution, with those EHEC materials, so ethyl hydroxyethyl cellulose. So those cellulose materials we've impregnated in that case with pure water. We didn't need glycerol because it was swelling, and yeah, we didn't need glycerol or DMSO. And so when we did that, we had a goal, and the goal was to measure how much of the C2 were substituted by the either ethyl or hydroxyethyl cellulose, or I should say ethyl or hydroxyethyl group, and which percentage of the C2 were not substituted. So this is the spectrum that we've published together. And you remember I said earlier that depending on the electron density, the chemical shift will be different for, let's say here, the cellulose, the C2 that is next to a hydroxyl group versus a C2 that is next to a substituted group. And so the C2 will have a slightly different chemical shift, whether it is substituted or not substituted, like here. But the problem here, like I said before, is that unfortunately the C2 area here is overlapping with the rest of the spectrum. So it's really hard to distinguish what part of this potato is essentially a non-substituted C2 and which part is a substituted C2. So because we don't have enough resolution, we need to go to a different type of experiment. And this type of experiment is called inadequate. The solid-state NMR spectroscopies, they really like to make some silly names for their experiment, because this one is actually very adequate for cellulose. So this type of experiment is essentially a carbon-carbon correlation. So on the x-axis, you have the regular carbon-13 NMR spectrum, but then you can separate in the y dimension, you can separate all the peaks. So the C3, C2, and C5 that were initially overlapping, here now you can see that are separated. C5 is here, C3 is in this region, and C2 is in that region. And now we can finally see that we have a different chemical shift for the C2 that is not substituted compared to the C2 that is substituted. And now if we want to know how much, we just need to integrate each of those two peaks, and we can get the percentage of the C2 that are substituted and the percentage that are not substituted. And this type of experiment, because it's a carbon-carbon correlation, remember carbon-13 is 1% abundant. So if we do a carbon-carbon correlation, we're only seeing the carbon-13 that are bound to other carbon-13. And this is only happening in 1% of the time. So we're only measuring 0.01% of the sample in that case. So this is why we really need the signal to be really strong, because we're really sensitivity limited here. This experiment took 24 hours with an enhancement of 120. This would not be possible at all if we didn't do DNP. We would need to acquire the signal for at least 6 months. And I'm really not joking. So we can just do some sensitive, well, actually insensitive experiments with DNP that we were not able to do before. Second case is, let's say you have hydrophobic cellulose, then the radical solution that you put will stay outside of those grains. And because the DNP effect is happening where the radical is, the signal will be increased where the radical is, so outside of the cellulose grain. But thankfully, there will be this diffusion of signal, so the large signal will be transferred to the inside of the cellulose particle by diffusion. It's actually called spin diffusion. It's essentially the same as thermal diffusion. But just for you to understand that this large signal provided by the radical will diffuse inside, even though the radical is not everywhere, the signal will go inside and be able to transfer that large signal to the particle. And we applied that for the dialcohol cellulose to be able to measure the degree of modification of dialcohol cellulose. So in that case, we only needed to do a regular carbon experiment with DNP, of course, because we wanted to have as large signal as possible. And then in that case, the C2 and C3 chemical shift of the non-modified cellulose was different enough from the one of the modified cellulose, the C2 modified and C3 modified. We could separate those peaks enough so that we only needed to directly integrate those different peaks. And then we would have to subtract the contribution for C6, C4, and C5, and just normalize those area. And then we directly get the degree of modification, or we tested 7 different samples. And we see that those degree of modification that are given by the relative areas of those peaks is very close to the degree of oxidation that was also independently measured by UV or titration. So this shows that DNP NMR is also a quite reliable method for determining the degree of modification of those dialcohol cellulose that are used for paint. And the last type of cellulose that I wanted to talk about is this HPMC cellulose, hydroxypropyl methyl cellulose, and it's also acetyl succinate in that case. So we have those cellulose backbone, and then the R group can be one of those. And in those cases, this hydroxypropyl methyl cellulose, there was actually a disagreement in literature in terms of the assignment of the peaks. So different studies were disagreeing on the location of the C2 next to an ether group on the carbon spectrum. And they were also disagreeing on where the C6 should be. They were not reporting the same chemical shift. So the first thing we wanted to do with this type of cellulose was to settle the debate and figure out which carbon was appearing where on the spectrum. And here, the same experiment as I showed you before, the inadequate experiment, this experiment, because it's a carbon-carbon correlation, the idea is that every time we have a peak that is aligned with another peak horizontally, it means that those peaks are bonded. So for example, it's color-coded so that you can see that easily. Let's say C2, C3 is the pink, and so you see that it's the pink here because carbon 2 and carbon 3 are bound. And so to settle the debate, the idea was just that since we know for sure that, for example, the C3 is around 80 ppm, and we know that the C3 peak is aligned horizontally with another peak, then it means that that other peak is C2. And so then it would mean that the C2 would be at 83 ppm. And so it means that it was this assignment that was the right one, and unfortunately, Pugliese was wrong about their assignment. And the same for C6. The C6 here, we know that it's this one because we know that C5 is that one and it's aligned with the other one. So this has to be C6. And so C6 is at 73 ppm, which means that Pugliese was actually right for the C6 ether and Zheng was wrong. So we settled the debate like that, but also what was really convenient with the fact that the hydroxypropyl methyl cellulose was hydrophobic was that you remember this spin diffusion of the signal to the inside. Well, the spin diffusion is not complete, meaning that if there is a cellulose molecule in the middle of the grain, it will not receive so much signal. So the enhancement of that cellulose will be a little bit lower. And this is what we picture here. So if we have some groups that are primarily at the surface, the enhancement will be larger compared to if those groups are in the core. And if they are homogeneously dispersed, then we see an average between some of them having a high enhancement and some of them in the core having a low enhancement. So by comparing the enhancement, we can know approximately whether the, let's say, modification is happening mostly at the surface, mostly in the core, or if the modification is homogeneous. And this is what we did with HPMC. We compared 3 different grades of HPMC, low, medium, and high, and we did the experiment 3 times each. And within each experiment, we showed the enhancement of the hydroxypropyl peak, the methyl peak, the acetyl peak, the succinyl peak, and the backbone is essentially the C1 peak, because we know that this one is just always the same, whether it's substituted or not. And we see that within the error of measurement, all the enhancements are always the same. The fact that they vary in between experiments is just due to the temperature that can sometimes vary. But what matters to us is that they are equal with respect to each other. And so because they are equal, it means that all those modifications, the hydroxypropyl or the methyl modification, they're happening homogeneously. So it means that they did a good job, because sometimes you think that the modification is everywhere in the grain, but actually it's only happening at the surface, because during the reaction, they only put the cellulose in contact with whichever product they used for the synthesis at the surface. And so the modification was only happening at the surface. So here we were glad to see that the modification was happening homogeneously throughout the grain. And there's another study from my colleagues who did essentially the same study than us. And they took 2 types of HPMC. They're called HPMC1 and HPMC2. They performed the experiment 3 times again. And they saw that for HPMC2, the enhancement between the cellulose backbone, so C1, the hydroxypropyl group and the methyl group, for HPMC2, they're all equal, meaning that essentially the substitution is homogeneous throughout the grain, which is exactly what we saw. But the second HPMC that was supposed to be the same, if you measure the enhancement, you see that the blue bars here are lower on average than the rest. So the hydroxypropyl groups has a lower enhancement, which means that the hydroxypropyl is mostly present at the core of the particle, whereas the rest is more homogeneous. Whoops. Go back. Yes. Whereas the rest, the methyl substitution is homogeneous. And so they could explain that even though they were supposed to be the same, they actually saw that there was different properties for both of them. And this property could be explained by the fact that those hydroxypropyl groups were only at the core for one of them, because hydroxypropyl is hydrophilic. And so if you concentrate it in the particle core rather than distributed throughout the particle, it changes the wetting property of the material, the swelling, the dissolution, the compaction behavior. It can change a lot of things. So with DNP NMR, we can answer those questions quite rapidly. And one last thing that I wanted to talk about, it's not a work that I've been doing, but it's a work that has inspired me. To remember this idea that this signal is transferred from the radical solution to the inside. Well, if you think about it, the part of the sample that is the most enhanced, so the part of the sample that has the largest signal, is the surface. So whenever you want to look at the surface in priority, if you who are listening to me, you have a cellulose material and you know that you are doing some modification at the surface and you want to primarily study the surface, you should really do DNP, because the DNP will make the signal the strongest at the surface of your material. And this is what some colleagues of us did. They did some phosphorylation of their cellulose, and the phosphorylation was only happening at the surface. And thanks to DNP NMR, they could answer a lot of different questions. The first question that they answered is that there was both mono and diphosphate group present at the surface. they knew this by doing directly phosphorus NMR, because you can do phosphorus NMR. It's 100% abundant. So if you have phosphorus in your sample, you can do DNP on phosphorus and you have large signals. You have a different chemical shift of the monophosphorus compared to a diphosphate group. Again, because the environment around each phosphorus is slightly different when it's a diphosphate compared to when it's a monophosphate. You can see that there is a little bit of diphosphate. You can even quantify how much diphosphate there is in your sample, at your surface at least. The second thing you can deduce is that the phosphate group are mainly installed at the C6, C2, but not C3 position. So you can see here that the C6, it has, this is the peak that corresponds to C6. And this is, as before, the peak that corresponds to C2, C3, and C5. And this is a correlation experiment, a correlation between carbon on the x-axis and phosphorus on the y-axis. And so you see a peak if this carbon is close to this phosphorus. So here we have a peak because we have a C6 close to a monophosphate group, which means that the phosphorylation is happening on the C6. And here as well, they managed to deconvolute those parts to say that the C2 was actually making a correlation between a monophosphate group. And so that's how they could say that the phosphate were mostly in C6 and C2 and not C3. And the last impressive thing that they could do is that when they looked closer at the C6 region, you can see that it's not just one peak, it's a convolution of a lot of different peaks. And actually, the chemical shift is not just dependent on the environment, it's also dependent on the conformation of the C6. So depending on if the C6 environment is trans gauche or gauche trans or gauche gauche, so this is the different rotamer that you can have, there will be a different chemical shift. And within each of those rotamer, you will also have a different chemical shift if this rotamer is phosphorylated. So you can deconvolute between phosphorylated gauche-gauche, gauche-gauche, and then gauche-trans, phosphorylated gauche-trans, and then trans-gauche. And then the phosphorylated trans-gauche is more to the left. And so by deconvoluting, they could even deduce what percentage of the glucose unit was in which conformation. So they really had so much information just by looking at carbon, phosphorus, and some correlation experiments of the two. So I think this is close to, yes, this is close to 40, 45 minutes. I would really like you to remember from this lecture that with DNP, we can do so much more than just regular solid-state NMR. There's a lot of questions that cellulose modifications are bringing, and a lot of techniques are struggling to answer those questions. And with DNP NMR, we can answer a lot of them. And I didn't even do a lot of cellulose. There's so much more things that you can do. You can know where the substituents are in the cellulose particle, in the molecule, how much chemical modification is present. That's what we saw with the dialcohol cellulose. And you can look at the surface modification of nanocellulose. That's what I just showed you now. And what is nice is that all of this that I showed you, you can do this with the magnet that I am playing with every day, which is here at the Swedish NMR Centre in Gothenburg. So if you want to analyze a cellulose material, you should really write me an email, and I'll be happy to design some experiments for you to be able to answer the questions that you have. Thank you very much. This is, of course, possible thanks to some postdocs that I've been working with that were financed by FibRe and Chalmers at the same time. and also the University of Gothenburg and the Swedish NMR Centre, who's paying me to do all this interesting and amazing research. Thank you.
Thank you a lot, César. This was an amazing presentation. We will see now if we can have any questions. Maybe even just think, yeah, there, I can stop my video. So thank you. Do we have any question directly in the chat? No, not yet.
No, not yet.
See here, Per, do you want to start or?
Sure, I can have one or two questions. First of all, César, thank you for a nice presentation. I have, I think, up to two questions at least, not necessarily related to NMR, but the effect of the result from the NMR, or how you can get more understanding. When you talk about the grains or granules of cellulose derivatives, since they are made originally from cellulose fibers and they don't really come in grains, how can one know that it's the distribution that differs, or that after the derivatization they have rearranged in different types of granules or grains, or you use the word grains?
Yeah, I use the word grains, but your question is, how do we know that the distribution of functionalization? So you're referring to this?
Yes, I mean, this is the interpretation of the NMR results.
Yeah.
Yes, but does that come from different reactions derivatization, or is this a redistribution of the polymers within the grains?
Yeah, that's a good question. That's actually a question that I was wondering myself. Unfortunately, I didn't get to have the answer from the manufacturers that were providing the cellulose to us. I wish they could. I don't even know if they know, but to answer this question, I think clearly what we could do is to do the same NMR measurement at early times, right after manufacturing, and then waiting for a given amount of time to see if there is this rearrangement of polymer happening within the grain, if that's what you're referring to.
Yeah. So it's more if you have any insight on, or is this a relevant question? But I from your
that's
answer,
a question that I
seems
personally
like
had, so it's relevant to me at least. But we didn't do those time point measurements, unfortunately. But yeah, that would be very interesting to do.
I don't know, are we receiving more questions, or should I continue with my second one?
You can take your second one.
Okay, so again, it's not the measurements you have been performing yourself, but in the end about the phosphorylation.
Yeah.
And I mean, general understanding is that the hydroxy group on C6 is the more reactive. So that's not surprising at all with the statement that they are primarily on the C6. But how did they interpret that they say that it's, or how do they motivate that it would also be why C2 is preferable over C3?
What do you mean they? You mean the
because you were not co-authoring this paper, right?
No.
This was a Japanese group.
Oh yeah.
So do you happen to remember how they did they sort of motivate, or why it might make sense that C2 is more reactive than C3?
Oh no, I haven't checked. I can check for you if you want. You mean on top of the NMR evidence, why would chemically make sense that C2
yes,
would be more
exactly.
reactive? No,
exactly
I haven't.
My question.
I haven't checked that. Sorry.
Because it's easy to argue that the hydroxy group on C6 is more reactive since it's a primary and so on.
Yeah.
But why would there be a difference between C2 and C3?
There are in the literature studies that clearly shows that C3 interacts very strongly with C with an oxygen on O5 oxygen, so along the chain. So
okay, so it depends on how that interacts with its neighboring.
yeah,
For
I didn't
the
read
EHEC,
exactly
it's the
this
same.
this
For the
paper.
EHEC, the ethyl and hydroxyethyl, they only go on the C2. I don't know how the reaction is performed. I just know that you don't see a C3 substitution. So there must be a reason why C2 is more reactive, and that's probably the same reason why it's present here in C2 and not C3. But yeah, interesting.
Yeah, that is a very interesting question. Here we have a question. I'm not sure who Josephine. I can't read fully. It's from Anna Ankarberg from AstraZeneca. So how do you know if you needed a glazing agent or not?
Yeah, glassing agent.
Yeah.
How do I know? Yes, good question. So essentially, when we record the NMR spectrum, the DNP enhancement is a very good indicator of whether we need a glassing agent or not. Because just by looking at it, when you see that, I mean, if it's super, super hydrophobic, you will see that your drop of water will not even enter your solid material. So then you don't even need to measure to be able to say, oh, maybe I need some glassing agent. And so but most of the time you put a drop of water and it looks like it goes in, but you don't really know if it's swelling or not. So you really need to measure by NMR. And usually what I do with the postdocs that I'm working with, we start without glassing agent. We just put the mixture of water and radical, and we look at the enhancement. If the enhancement is in the order of higher than 50, between 50 and 150, it means that the DNP effect is working great, which means that the matrix is for sure glassy, which means we don't need glassing agent. And we will see it very quickly if the matrix is crystallizing, because the enhancement will be 1 or 2. So in that case, we need glassing agent. Another way of seeing it is, so on your carbon spectrum, you have a peak that corresponds to your solvent. Let me see here. Here, this peak. The width of this peak can tell you whether it is crystalline or amorphous. If it's crystalline, it's sharp. If it's amorphous, it's broad. And so if it crystallizes, it means that your peak will be sharp, in which case you need a glassing agent because it's crystallizing. And if your peak is broad, you're probably good because you have already an amorphous matrix when you freeze, so you don't need that glassing agent. So both a good enhancement and broad peak is usually a sign that everything is amorphous as you want and you don't need a glassing agent. And if you have low enhancement and sharp peak, you probably need a glassing agent.
I think Anna was happy. Anna, do you have any more questions? Then we can let you in directly, and you can ask them directly. In meantime, I can ask a question about you showed the effect on where you could see if the substitution was on the surface or in the middle for the HPMC and HPMC AS. If you go back to the alcohol cellulose samples, did you saw anything similar there? Could you see if they were similar substituted, or maybe you didn't have time to check that?
Oh yes, here, yes, I didn't show it to you, but so we use another method to determine whether the substitution was at the surface or in the center. We use, it's called the build-up time method. You compare different build-up times, and if they're all identical, it means that it's homogeneous. In that case, all the build-up time were identical. So we were able to suggest that the modification, this dial called cellulose modification, was homogeneous. I didn't show it because it uses another method, and I would have had to explain it. But yes, homogeneous.
And how the fibrils are in the dimension. You can have a long discussion about how big a fibril is. Per can help me here, but say 3 nanometer or something like that. Maybe a bundle a little bit larger. Do you think, does it mean that it's homogeneous in the whole fibril, or what does it mean? Or it can the signal be if you have so small units that they are
Yeah,
you get the signal that it is uniform?
Yeah, it's a good question. So this this diffusion of signal, it's usually in those cellulose material, it's usually tens of nanometers or maximum hundreds of nanometers.
Mm-hmm.
So if you want to be able to look at a difference in substitution within, let's say, 3 to 5 nanometer, DNP is maybe not the method that will give you the best contrast for that. It's just that when we have typical grains, if they are on the order of micrometer or sub-micrometer, let's say several tens or hundreds of nanometer, that's perfect, because then it corresponds to roughly the length scale where diffusion is limited. And that's where you're going to be able to say, okay, if all the enhancements are the same, then modification is the same within that hundreds of nanometer length scale. It's always, when we say homogeneous, I should specify to which length scale. You're right. But so less than 10 nanometer, I don't think we can be confident enough to say whether there is a heterogeneity within that length scale. I think it's a little bit too small for us.
Okay, yeah. Thank you for that answer. Josephine, do we have any questions in the Q&A in the
No,
YouTube?
I don't see it. No, I don't see any more questions.
I don't know. We are 3 minutes left, so maybe I need to stop ask questions now. But I would really thank you, César, for a very nice presentation, and it was very interesting and pedagogical. I think I almost understood everything, so that was fantastic.
Yeah.
So thank you a lot, César, for this effort to put this together.
Thank you very much for inviting me. It's a pleasure. And yeah, if any of you listening wants to study cellulose, you have my contact email. I'll be sure to do the best for your cellulose material.
Thank you. That sounds good. We have always a lot of questions.
Yeah. That I know. The questions are not missing. They're not lacking. It's really interesting.
So yeah, thank you. With that, I think we give the word to Josephine. We will have a new webinar in the autumn coming together with FibRe and Treesearch. So we are looking forward to that, Josephine.
Yeah, I don't have much more to say. Thank you for you who attended. And if you want to see any recording of the previous webinars, you can find it at the webinar webpage or at YouTube. So if you missed some, or want to watch it again, you can always go back there.
Absolutely.
Thank you very much, everyone, and thank you for the presentation.
Thank you, everyone. Have a great day.
Yeah, the same. Thank you.
Bye.
Bye.
Bye-bye.
Thank you.
Då Josefin